Today, 22nd January 2022, MLT second year, subject is microbiology. Topic chapter is mycology. Topic is dermatophytes. Dermatophytes are a group of morphologically and physiologically related molds. So dermatophytes are physiologically or morphologically. They are molds. Next point, dermatophytes are a group of filamentous fungi. Dermatophytes are group of filamentous fungi. Some dermatophytes are causes for skin and hair and nail diseases. Some dermatophytes are causes for skin, hair and nail diseases. Some some dermatophytes are causes for ringworm diseases. We have to see in advertisement in TV channels, rich guard, ring guard. So they are used for that tubes, that creams are used for dermatophyte fungal infections. Some uh, some dermatophytes are classified. One is Trichophyton, Microsporum, and Epidermophyton. Trichophyton, Microsporum, and Epidermophyton. These are three types of consist of dermatophytes. This type of dermatophytes are hyphae or arthrospores. Dermatophytes are hyphae and arthrospores. Cultural characteristics in dermatophytes are growth on Sabrodoxos agar. Sabrodoxos agar is a fungal culture media. So, especially these fungal culture media means culture media consists of nutrient agar, blood agar, chocolate agar. A number of medias are there, but especially this type of Sabrodoxos agar only for fungal culture media. Why? Because this agar consists of cyclohexamide and fungal infections. So this type of micro or fungal organisms, they are growing in this type of media. So uh, some trichophyton and microspora and epidermophyton, these are species in especially they are going into dermatophytes. So some trichophyton species and microsporum species and spindle shaped species, this type of species are used for dermatophytes. Trichophyton colonies are powdery, velvety, or waxy with characteristic pigmentation. Microconidia trichophyton species or colonies are powdery, velvety. In microconidia are abundant, arranged in clusters. Macroconidia are scanty. They are elongated with blunt ends and distinctive shapes. Some species possess in hyphal characters. Such spiral hyphae, rocket mycelium and fabic chandlers. Trichophytons infect skin and hair and nails. Trichophyton rubrum is the most common species. Microsporum colonies are cotton like velvety or powdery. Microsporum. Trichophyton species are powdery and velvety pigmentation. But microsporum colonies are cotton like velvety powder or powdery or brown pigmentation. These are microsporum or brown pigmentation. Microconidia are scanty. Microconidia are spindle shaped in microspora. Some microsporum species large, multicellular, and singly on the ends of hyphae. On the ends of hyphae, singly multicellular, and this is the microsporum species. Microsporum infects hair and skin and nails. Microsporum species are especially they are causes for nails, hair and skin. Epidermophyton. Epidermophyton colonies are powdery with greenish yellow color. Colonies are greenish yellow color. Microconidia are absent. Microconidia are absent. Macroconidia are multicellular pear-shaped arranged in clusters. Macroconidia are multicellular shaped and arranged in clusters. It is infect to the skin and nails and hair, not in hair infection, especially infect to the skin and hair, nail. Example, Epidermophyton flacosum. In lab diagnosis is made by examination of potassium hydroxide. Uh, how to diagnosis in laboratory skin samples and nail clippings and hair clippings. So you have to each sample you have to collect, especially skin samples you have to scraping. After scraping, they are placed in the slide. Then potassium hydroxide by one drop of potassium hydroxide. After 15 minutes, in dissolving them, you have to see in cover slip and see in the microscopic examination. Same as it is in hair infection and nail infection also. But sample collection is differently to the skin infection and hair infection and nail infection. So this is the very important, especially this used for 10% KOH in hair infection, skin infection and nail infection. So a cover slip is placed in the preparation of gently heated bring the about clearing. In laboratory, microscoping shows branched hyphae, septate hyphae. So, uh, all type of fungi they are seeing in the septate hyphae, branched hyphae, unseptated hyphae. These types are used in the microscopic examination. So, next topic is Aspergillus. Aspergillus is a morphology short, swollen, filamentous, and swollen tips. Their pellets should be small and hard. Smooth surface. This, this is the morphology of Aspergillus. So, what is the cultural characteristics of the Aspergillus? Aspergillus species is a highly aerobic and found in almost all oxygen rich environments. Highly oxygenated areas they are growing in the Aspergillus species. So, where they commonly grows as molds on the surface of a substrate as a result of the high oxygen tension. Commonly, fungi grow on carbon-rich substrates like monosaccharides. So, some Aspergillus species are common contaminants of starchy foods and grow in or many plants or trees. In addition to growth on carbon sources, many species as Aspergillus demonstrate in oligotrophy. So, uh, some species are consist of Aspergillus, Aspergillus niger, Aspergillus flavus, uh, and some Aspergillus is a group of caused by aspergillosis. The disease is the name of aspergillosis. The most commonest subtype of aspergillosis, Aspergillus fumigatus. Especially the most commonest fungal disease in Aspergillus is called Aspergillus fumigatus, underline inverted commas. So, what is the symptoms of Aspergillus fumigatus? Like the patient consists of fever, cough, chest pain, and breathlessness. So, these symptoms are especially used in Aspergillosis patient. So, what is the laboratory diagnosis in Aspergillosis patients? Diagnosis may be you have to which sample you have to collect means especially Aspergillus flavus means is you have to take the sputum sample. You have to take the sputum sample. That sample consists of a you have to take the separate dextrozagon. They are growing in the Aspergillus species. You have to isolate from the potassium hydroxide KOH mount. You have to do the KOH mount, especially used in the in this laboratory diagnosis. But how to culture in Aspergillus fumigatus? So you have to, especially they are growing in the Sabra dextrose agar on culture media in 25 degrees centigrade. So what is the colonies are velvety, powdery surface or colored are seen, especially this conformation of the Aspergillus fumigatus are seen in the Sabra dextrose agar in these patients. So next continuation with the penicillium. Thank you all of you.